So thank you all for joining us to this RedTalk web conference organized by the Institut Carnot Calim. And I would like to thank all the Calim RedTalks team for their commitment to make this event a success. So my name is Karin Tarte. I am an immunologist by training, working in Rennes in France on lymphoma microenvironment. And it's truly a pleasure for me to share this session today with my friend and colleague, Sandrine Roulland, from Marseille, and to open the second episode of the RedTalk Season 6 on a fascinating and very timely topic, preclinical models, organoids versus animal models. And we are fortunate to welcome two experts in this field who will each present their work for about 15 minutes, followed by a few burning questions. But we'll save the best for last, an exchange that we hope will be dynamic between you, the audience, and our amazing speakers around all the questions you may have on this major topic. So feel free to share comments and ask your questions in the chat, making sure, or in the Q&A box, making sure to indicate who you are and take this opportunity to be actively involved for everyone's benefit. Sandrine.
Yes, thank you, Karin. So my name is Sandrine Roulland. I am a research director at INSERM in France, working at the Center of Immunology and focusing on B-cell lymphoma pathogenesis. So it's really a great pleasure today to welcome our two speakers, Professor José Angel Martinez-Climent and Professor Elisa Oricchio. So Professor Climent, who will start, he's a leading expert in hemato-oncology coming from the University of Navarra, and he has developed a large part of research creating genetically engineered animal models to uncover the molecular mechanisms of lymphoma and myeloma genesis. And today he will present us the zoo that he has constituted and how we can use those tools to explore the mechanism of response of immunotherapies, etc., etc. And so it will be the first presentation, and the second presentation will be by my dear also friend and colleague, Professor Elisa Oricchio, who is coming from the École Polytechnique Fédérale de Lausanne in Switzerland. And so she is an internationally recognized researcher in lymphoma biology who has been focusing a lot on understanding the 3D chromatin structure in linking to oncogenic pathways activating in lymphoma. And she has also developed a platform of patient-derived 3D ex vivo model in order to study the tumor microenvironment interaction and also predict therapeutic response. And so together they will really offer us a perspective on how these two models can be used and can be leveraged either individually or in combination across preclinical and translational research. And so now I think it's time to give the floor to Professor Martinez-Climent for the first talk. Thank you very much for joining us.
Thank you very much, Sandrine, for the kind invitation. Thanks to the organizers. I'll do my best to keep in time, right? So I was telling that one of the major questions we have in the field is to decide which among the many different models that we have is the more appropriate to study human cancers, ranging from xenograft models, PDXs, genetically engineered to syngeneic transplantation models. What we've been trying to do in the laboratory during the last years is try to use these mice to address particular questions that we may have, clinical questions, scientific questions. And to do that, we've been making these models more like humans. Next slide, please. I will tell you more about the process that we've been doing, the humanization of the different mouse models of multiple myelomas, leukemias, lymphomas. Today, I will be talking about multiple myeloma. Next, please. So we published several years ago this collection of myeloma models coming from genetically engineered mice, where you can see in the slide that we think we are reflecting in these particular models the principal pathogenetic elements of the disease, including the genetic heterogeneity, but also the progressive transition from early stages into clinical active disease, and very importantly for us, the interaction of tumor cells and immune cells during the progression. Next slide. In these models, we learn a lot of things from genetic analysis of tumors, but we were very happy to show that in myeloma models, in patients as well, we can characterize the immune system in the bone marrow. Next slide, please. Next. And divide, no, sorry, previous one. I'm so sorry. and the previous, and divide the disease into what we call, depending on the tumor microenvironment, immune microenvironment, immune color, immune inflame. And what we are now learning is which are the different genetic lesions that drive these immune inflammatory conditions versus the colder conditions. This is very important. Next slide, please. Not only because we learn a lot from that, but also, next please, because having a look and characterizing in deep detail the immune microenvironment in myelomas, in patients and in mouse models, we are able to predict which of these tumors is going to be more aggressive in comparison to the others, and also, very importantly, how the immunotherapy responses are going to be in mouse models and in patients. So, yes, next slide, please. So what we do in the laboratory is to run this preclinical therapeutic analysis or studies. On the left side, you can see a couple of major survival curve testing 2 different treatments for myeloma, lenalidomide in comparison to a triple therapy, which is standard of care for our patients, including bortezomib, lenalidomide, and dexamethasone. You can see better responses in the VRD arm as expected, but we not only get longer survival in the triple therapy in comparison to lenalidomide, but we can also measure sequentially the dynamic responses across time. And in the slide, you can see that measuring the gamma fraction, we can, and also the number of myeloma cells that we can measure directly through bone marrow aspirates. With a very simple flow cytometry, we can characterize the deeper responses across time. And you will see there in the slide that at week number 8, after treatment with the VRD, we get a complete remission, which is very similar to what we can observe in our patients with myeloma. But if we keep treatment, we keep following up these mouse models, and the number of myeloma cells increase in the marrow, getting a relapse of the disease, and eventually all mice die with a relapse of myeloma. This is what we call a refractory state in our mouse models that we think reflect very much the clinical evolution in our patients. And if you move, please, to the next slide, just to show you that also genomically we can characterize refractory states versus the newly diagnosed myeloma. In the slide, you have a couple of examples, and the number of mutations, the genetic complexity of the relapsed myeloma are in line with what we are familiar in patients. And these genetic lesions that we know occur in the relapse state, P53, 1QM, and so on. Next slide. So once we have this complex model of relapse, we can address questions that are not possible to be responded in other models, and of course, not in our patients. We can do preclinical therapeutic trials where we can combine, we can compare any treatment. In this particular slide, we did something that is now being compared in the clinic. We just treated our myeloma models with a BCMA CAR T cell, which, as you well know, is approved to treat our patients with relapsed myeloma. And there are now clinical trials trying to demonstrate that these same CAR Ts are also effective and maybe curative in first-line treatments. And the answer, according to our mouse models, is that maybe we have much better responses. You can see in the slide in the newly diagnosed myeloma models in comparison of to VRD treated as myelomas, relapsed myelomas. Next slide, please. Going back to the model, we also were able to delineate these complex minimal residual disease stages that is very difficult to study in patients. And also, I will not tell you about all the characterization of these MRD cells, but we'll tell you that we also apply some preclinical therapeutic trial. Next slide, please. Where we address something that is really in our minds for many years. Can we treat our patients with myeloma before relapse, which is what we have been doing on the clinic for years? And the answer is we can treat at the MRD state. In this case, again, is the same BCMA CAR T that can be administered in myeloma models after VRD treatment. And then at the complete remission stage, we randomize those mice that receive at that minimal residual stage the CAR T while we wait until relapse and administer the same CAR. And responses are dramatically different. In the red line, you can see a major sustain in the relapse. In the minimal residual state at the complete remission, and also it is reflected in the survival on the in the increase in survival in comparison to the late stages. Again, we can test these experiments that we cannot do in patients in our mouse models and address questions that guide our clinical decisions. Next slide, please. I will keep share. Next slide, please. Next, please. Yeah. All right. So the next step we've been doing in the lab for the last years is to humanize the targets that we are interested according to our immunotherapy agents in the clinic. CD38, that's LAMF7, BCMA, GPRC5D, FcRH5 are targets where we have now monoclonal antibodies, T-cell engagers, CAR-Ts in the clinic, and we have humanized all these particular genes in the mouse cells by replacing the endogenous mouse gene by the human gene, right? In order that we have a mouse model which is completely mouse, but just the extracellular domain of these targets is human. And also we did the same in the T cells in these particular mouse models, and therefore we are able to test directly the same immunotherapy agents that we are testing in the clinic in our mouse models. And I'll show you a couple of examples of these sophisticated animals. Next slide, please. This is one of the myeloma models that I showed you before. The difference is that here we humanize BCMA and CD3 in order to make the murine cell sensitive to the T-cell engager, but also cereblon in order to make the cell sensitive to IMiDs and the new cell months. Next slide, please. This is just a preclinical therapeutic trial where we combine a T-cell engager at a low dose and a high dose, and we observe the same results as we are seeing in our patients in the clinic. Major major increase in the survival time. You can see in the Kaplan-Meier survival curve, but this increase in survival is associated with toxicity, with immune toxicity, in particular T-cell exhaustion, but also the secretion of interleukin-6, interleukin-6, and many other cytokines that induce CRS in our patients. So we are recapitulating not only the mouse model and the myelomas, but also the responses that we see in the clinic. Very good responses with immune toxicity, which is a major problem in the clinic. And we use, next slide, please. And one of the projects that we have done and was presented a few weeks ago by my colleague Marta Larrayoz in one of the myeloma meetings in Miami was that we try to mitigate these immune toxicities using these humanized models. And in the slide, you can see that just by combining with one of the drugs developed by a company, which is mezigdomide, which is one of the new cell mods, right? We were able, next please. We were able to keep these huge responses, but at the same time, without, next please, and without entering into the details, we were able, as I mentioned, to keep these huge responses as an increase in the survival of the mouse models without inducing T-cell exhaustion, in particular to the CD8 T lymphocytes that did not acquire the expression of the markers of exhaustion, TIGIT, LAG3, TIM3, and so on. And also, and finally, and at the bottom of the slide, you can see that there's the secretion of these interleukin-6, interleukin-10 cytokines associated with CRS is mitigated. Therefore, we are modeling a new treatment for these T-cell engagers in combination that mezigdomide that can be very effective in our patients with myeloma in the clinic. Next, please. We also were interested in the mechanisms of these combinations, synergistic, not only in the efficacy, but particularly in the immune toxicity. Making long stories short, we apply single cell RNA sequence analysis and observe a major expansion of CD8 T lymphocytes with an stem-like phenotype with expression of LAMF7 cell and so many others in the treatment, in the combo treatment, in the combination treatment, in comparison to the T-cell engager immunotherapy, which we think are those T cells responsive for the phenotype in the mouse models. And very importantly, these CD8 T lymphocytes with expression of stem cell markers did not express as TIGIT, LAG3, TIM3, and so on. Next slide. So in order to validate this functionally, what we did finally is to combine a T-cell engager and with the monoclonal antibodies targeting these immune molecules, PD-1, TIGIT, LAG3, TIM3. To do that, we use a syngeneic transplantation model, which is not as complex as the one before. And here we could do a lot of experiments in combination in a short period of time. You can see in the slide, just by the combination of the T-cell engager, the BCMA-CD3, with an anti-LAG3, we could see an increase in the survival. But we keeping the combinations going in this simple model, next slide, we were able to show that by combining the T-cell engager with an anti-PD-1 plus anti-LAG3, we got beautiful extension in survival in this particular mouse model of multiple myeloma, again suggesting us that these preclinical studies can guide our clinical decisions, and maybe we can think in the field about including these molecules, anti-PD-1 and PD-L1, into the myeloma field. Next slide. Finally, we have also humanized one of the targets, GPRC5D. I will be very, very brief here. We not only get beautiful responses that I won't be showing you today, but this T-cell engager that is in the clinic to treat our patients with multiple myeloma is associated with immune toxicity, but also with non-immune toxicity. And this is very painful for the patients, particularly the skin rash that they have after treatment with this molecule, and problems in the tongue, dysgeusia in the nails, and so on. And the reason to do because of that is that GPRC5D, which is a marker, which is a molecule expressed almost selectively by the myeloma cells and by the plasma cells, is also expressed by a subset of epithelial cells. Next slide. Unfortunately, in the mouse models, GPRC5D is not expressed in the myeloma cells, but making a long story short, we humanized GPRC5D and were able to have a model with a humanized GPRC5D expressed in the epithelial cells in the skin and the tongue. And after treatment with a T-cell engager, the same T-cell engager that we are using to treat our patients in the clinic, we were able to reproduce the non-immune toxicity in the skin and the tongue and in the And in the slide, you can see a major infiltration of CD3 T lymphocytes, particularly CD4, and surrounding the epithelial cells with the expression of the human GPRC5D, which we think is the cause, which we think is causing this non-immune toxicity in our patients. And we are now interested in deciphering the mechanisms of these T lymphocytes, and in order to find a solution to treat these patients in the clinic. And I think I will stop here. Next slide. Yeah, I was going to tell you about PDXs, but because I'm so late, just next slide, I think is the last. Just to tell you that we have been able to develop PDX models of multiple myeloma from relapsed patients, and we're trying now to humanize the immune system in order to also conduct these immunotherapy trials in our mouse models that are so informative and are helping us to advance to the clinic. Next, please. My final slide. Next, please. Next slide. Yes. Yes, just to show you that there's no right model for all experiments. We need to decide which is the best model we have for each particular experiment. Of course, I'm talking today about mouse models. I know that Dr. Oricchio next to me will be talking about organoids. So we have to decide which among the many different mouse models, organoids, PDXs, and so on we have in our laboratories in order to address our particular clinical scientific questions. And I'll stop here. Again, I'm so sorry for for the inconveniences, and I'll be happy to discuss with you and address all questions at the end of the session. Thank you so much.
So thank you, José, for this nice presentation. I think, is there any burning question here, or should we, maybe we can go directly to the next presentation, and then we will have a final, I think, final discussion and interactive question between the two presentations, so that we don't lose the flow, if it's okay with you. So Elisa, if you want to share your slide.
Okay, I hope that you can see the slides and see me. I think it's, first of all, thanks so much for organizing this session and having this debate. I have to first admit that I love mouse models, so I'll do the devil advocate, but we will see at the end how we can place the two models together. Just to tell you a little bit, to introduce myself and what we do in the lab. Let's see if the slides. So the main area of research of my laboratory has tried to capture, let's say, tumor evolution in a sense where we can look at normal cells and how they transform and become tumor cells. But in this, we know that the cancer cells do not live in silos. They are a constant interaction with the tumor microenvironments. And so in the lab, on one side, we are trying to understand cancer cell intrinsic alteration, so how genomic alteration can influence the three-dimensional structure of the chromatin in the nucleus and how this contributes to tumor development. But at the same time, we are also interested in understanding the communication between the cancer and immune cells, and specifically on this context, because we think that here we can either identify new therapeutic targets or improve therapeutic treatment. So it's a bit more on the translational side. And specifically for this, let's say, communication between the cancer and immune cells, the model that we study the most in the lab, it's of course lymphoma. I think for this audience, probably the introduction is too simplistic. I just put here a few key points that we want to highlight. So that first of all, we are looking at an extremely heterogeneous disease. So we call a lymphoma a lot of different types of malignancy, where the cell of origin is actually different. So we have lymphomas that originate by B cells. They are in different stages of differentiation. And on top of this, we have a significant number of genetic modifications that contributes to the tumor development. At the same time, even if we are looking on the same lymphoma subtypes, and here I highlight diffuse large B-cell lymphoma, in the same subtypes, we have multiple different genetic alterations that contribute to different evolution of the disease. So we have different cellular origin, different genetic alterations, so genetic heterogeneity. On top of this, we know that lymphomas live in secondary lymphoid organs, and they have a different dependency on the tumor microenvironment. For example, here we have a picture of follicular lymphoma. We know that this is an indolent tumors that strictly depend on the interaction with the CD4 T follicular helper cells, follicular dendritic cells that are stimulating the proliferation of this disease. However, we know that the follicular lymphoma can also transform in a double hit aggressive lymphoma through acquisition of different genetic lesions, like, for example, MYC overexpression. And this not only changed the genetic of the disease, where we have that cells become larger, but also completely change the interaction with the immune microenvironment, where we have an effacement of interaction with the CD4 and other cells, and we mainly recollect the presence of macrophages. So just to summarize, what we have here is that probably response to therapies in lymphoma needs to consider all of these different dimensions. It can vary based on the stage of the disease, the type of genomic lesion that we have, and also the level of immune cells infiltration. So the question that we had in the lab, how we can actually recapitulate this. And of course, we know that we can use cell lines. Human cell lines are great to recapitulate heterogeneity of the genetic of the disease. We don't need to discuss the limitation of cell lines. We saw the beautiful presentation of José describing the animal models that we can generate, and they are great to recapitulate the systemic level of the disease. But sometimes it's difficult to mimic the genetic heterogeneity that we normally can see in the patient, because we need to breed and cross this animal to actually have this genetic heterogeneity. And more, we started to reveal the importance also of the epigenetic heterogeneity that we have in the patient. The PDX are great, but sometimes are limited for indolent lymphomas, and we know that they lack the tumor microenvironment. So we started to think whether we can have new models or different models that can help to address at least a part of the problem that we have. And for this, we want new models to address, let's say, two questions. On one side, we want to fast assess therapy response, and also in a context a little bit of personalized medicine, try to see which therapy is the best for each patient. And on the other side, we want to try a model in which we can study lymphoma evolution, where we can maybe detect lymphoma initiation that is a little bit more difficult to detect in the patient, because we only see the patient when the tumors is actually already there. And so for this, we came up with this idea to develop a new models that we call lymphomoids. So I'm going to tell you a little bit about this model. So this lymphomoids, it's basically an ex vivo culture system where we take secondary lymphoid organs, either for the mouse, or if we can obtain fresh tissue biopsy from human. We isolated the tissue, we fragmented the tissue, and we have created this synthetic system that it's air-liquid interface base, where basically the tissue fragments are in this chamber that is surrounded by the media, but not indirect contact of the media. So there is a gradient of nutrient here that mimics a little bit better what is the normal physiological condition. So we started generating this using murine models. And first what we did was to compare, after a few weeks in culture, how the architecture of the tissue was maintained. This is the original tissue of a spleen of a mouse that present initiation of follicular lymphoma. And this one are a lymphomoid that has been generated from the same animals and maintained in culture for a few weeks. And we can see that we still notice the persistence of these lymphoma cells that are accumulating here. We can also use this system to study basically the 3D structure of the tissue. What is important here, that we don't disrupt, it's not organoid, we don't disrupt the tissue, we really maintain the physiological interaction between the lymphoma cell, for example, CD3 T cells and vessels. And we can also see that overall, in a period between 2 or 3 weeks, the overall survival of the cells is good. We have more or less maintained the B cells population. We can recognize the presence of some germinal center B cells, T cells, and CD4, CD8 cells. So overall, although not perfect, we thought that it's actually a nice model that we can try to test whether this could be useful in a clinical setting. And so on this, we team up with the hematology department here in Lausanne. And so the head of the lymphoma unit is Dr. Anne-Carolie. And then Justine Epinay was an MD PhD student in my lab that contributed to create the liaison with the clinic. And Albert Santamaria Martinez is the postdoc running this project. The idea was to actually collect fresh lymphoma tissue that we then took in the lab, reduce this tissue in small fragment of 1 millimeter cube, and then use this tissue to test different therapies in parallel. And then we can do an image analysis to see which drug would be effective on this patient, and then compare the response with what happened to the clinic. For this, we have done a first study where we had collected 27 different lymphoma biopsy of a different subtype of lymphoma. And after a quality check, based on the amount of material that we receive from the clinic, we test between 4 to 7 different treatments. And so here it's a summary of few results. So first of all, we compare the original tumor with the lymphoid that were in maintaining culture for a short period of time to see whether we can maintain and correlate the number of B cells, CD4, CD8, and macrophages, but also the level of proliferation that we normally detect in this tumor. We also perform some spatial transcriptomic analysis to see whether on this lymphoid we maintain the heterogeneity that we see in the original tissue. So this is a biopsy of lymphoma, it's a needle biopsy. You can see that there are areas that are rich in tumor cells that have this red dot here, and others that are mainly rich in stroma cells. And these are the lymphoid that we have generated, so 6 different ones. And here we can see that we recapitulate the heterogeneity that we see in the original tissue, where some lymphoid are more rich on tumor cells, while others are more rich on the different component of stroma cells. And at this point, we start to compare therapy response. We mainly use a small molecule inhibitor. And so here are a few patients, and here is the summary. And for each patient, we could see which therapy was give a preferential response, and we are measuring proliferating B cells in terms of therapy response. And for example, we have a couple of patients where none of the therapy that we tested actually worked. And for a few other patients, we could see that, for example, they have a preferential response either for idelalisib or for ibrutinib. In this, we could also try to understand what was this difference between these different patients. So because we have the entire tissue architecture here, we could do spatial transcriptomic analysis on lymphoid untreated and treated with the drugs. And first of all, we can start to see if we look at H&E of this lymphoid, that in the untreated one, we had several areas that are rich in tumors. With ibrutinib, we could not recognize any tumor cells, while with the idelalisib, we still have the presence of tumor cells in this lymphoid, suggesting that in this case, we have a preferential response for ibrutinib. Interesting, when we saw why and what are the genes that were differentially expressed in this condition, we noticed that in ibrutinib, we have a significant upregulation of immunomodulatory molecule. So the important part here is we don't only see response of the tumor cells, but we actually can trigger a response of immune stimulation. And indeed, if we look at the details of different population that we can identify on this lymphoid, and we compare ibrutinib versus untreated tissue fragment, we notice that not only we reduce the number of tumor cells, but also the number of B cells, but in the culture, we can start to see an expansion of CD8 T cells, some macrophages, and dendritic cells. So the idea that in this model, we can actually start to understand the interaction between the cancer and immune cells upon therapy response. Then the last question was, is this relevant? Can we actually use in the clinic? And for a really limited number of cases, what we had is matching what happened in the clinic with what happened in the lab. So I just show you a couple of examples. So this was a patient with small lymphocytic leukemia. We received the biopsy and we treated with ibrutinib. And so we could see that there was a good response. It was expected. So most of the patients respond to ibrutinib. And so we could confirm these results in the lab and also in the clinic. What we also noticed that in some cases we can anticipate whether a patient could be resistant. This is a patient that was diagnosed with a non-germinal center DLBCL. This patient was treated with lenalidomide, and we received the biopsy, and after 7 days treatment with lenalidomide in the lab, we saw that there was no response. And the same happened after a few weeks of treatment in the clinics, where actually the tumor progressed and we have the emergence of additional lesion. So this is relatively a small case study where out of, we received 9 cases where we had match response, and 8 out of 9, we observe a similar response in the clinic and in the lab, suggesting that potentially this can be a platform that can be applied or used in the clinic to try to understand whether a patient will benefit for some therapy. And just to quickly tell you that, of course, we started this in lymphoma, and then there is also a general interest now to see whether this could be actually a platform that could be used also for different tumor type and solid tumor, and be coupled with a molecular tumor board decision. And for this, we are expanding this platform not only to lymphoma, but also to other tumor type, for example, in lung cancer, on the glioblastoma, to actually try to help decision-making in the clinic. And we have created a network. This is the French side of Switzerland, where we have different hospitals that are seeing different tumor types. And so we are collecting these tumor types to actually see whether this platform can be really informative at the clinical level. Now, just in the last minute or so, I told you, okay, this is one application where we can see response to therapies. There is also an interest in the lab to try to better understand lymphoma initiation and its evolution. And we wonder whether we could use this lymphoid to start to address this question. And so we had basically used mouse lymphoid that we started to engineer with an optogenetic system where we can induce with light and started to study lymphoma initiation ex vivo. And I'll just show you some preliminary data that we have done on this as a proof of principle, where we basically take VAV-PCL2 mice, we collect hematopoietic progenitor cells, and we have engineering plasmid where upon light induction is switched to green to red and induce the expression of oncogene. And so this is a project that is pioneered by us, a PhD student in the lab. So when we obtain a chimeric animal models, we collect the spleen, we chop the spleen in a small fragment, creating a lymphoid. At this point, we have in this lymphoid a really small percentage of cells that are actually recognizable because they are red. Then we are exposing to blue light, and this activate a Cre recombination that induce a switching color, and this is associated with activation of MYC. We can see the initiation of the tumor because the cells are green, and then look at how these cells actually form tumors. And this we are combining this platform with live imaging of 2-photon microscopy. So this one is one of the first experiments that we have done. So I hope you can see there are really few red dots that we can see here. Then we can illuminate this tissue, and then we can start to basically track 8 days post-illumination when we have no oncogene, or if we started to express MYC on these cells, how this clone expand, and the number of positive cells that grow over time. So we think we start also, of course, to see whether these cells can be oncogenic. So we can take these cells, retransplant in animal models, and then see whether or not they actually can form the tumors. So I think it's just that we think that with this tissue explant, we might be able to address or to use as an alternative to mouse model to understand how different genetic lesions evolve. Maybe they are a bit easier to engineer and to obtain combination of different genetic lesions. In this tissue explant, we have the full tumor microenvironment, so we are not changing the composition of the tumor microenvironment. Can we actually be used in a clinical trials or clinical routine to identify what is the best therapy for each patient? So, of course, open question. And maybe the big one for this audience, can actually this replace mouse model? And probably this is a provocative question that I will close the talk with this. And thanks the people in the lab. I already mentioned a few during the talk, and I think we can start the discussion.
Thank you very much, Elisa, for this very nice presentation. We have a lot of questions from the audience. Perhaps I could start with questions from Christine Bezombe in Toulouse, who probably know we developed with Camille Laurent the PDLS model. So she has several questions. The first one, perhaps, did you apply T-cell-based therapies, and can you follow T-cell activation in your model? And if yes, where within the lymphomas, where are these T-cell activation take place?
Okay, so we tested in terms of immunotherapy, we have started to test the CAR T cells. So introduce CAR T cells and see whether they can infiltrate in the lymphomoids. So there is some infiltration, but it's minimal. They tend to stick a little bit at the periphery, and so we are trying to change a little bit the matrix to see how we can improve this infiltration of T cells. So we can discuss a lot about what are the limitations of this system, and I think there are several. It's maybe just at the beginning. And what I found that the most challenging part of immunotherapy response and T cell activation, that it really depends, it's a static system. It depends on the resident cells. We are not recruiting anything from outside. And so this is a limitation. And we are trying to address this. So for immunotherapy, I think we can address some, but maybe we should also acknowledge the limitation that we have there.
So a question that is related probably to this one is another question from Frédéric here from Rennes, Frédéric Moursin. Did you check whether your lymphoma model contains fibroblastic cells that not only provide survival factors, but also biomechanical constraints or produce chemokines and so on that could affect drug diffusion and cell recruitment also?
I think, yes, we have a fibroblast. We have done a staining with some Luna for this spatial transcriptomic, or spatial proteomic, and we have a fibroblast that are present there. We are also able to isolate some fibroblasts from this culture if we dissociate. So we have to characterize what are these fibroblasts, if they are, what type of fibroblasts. So we think that might be some, different type of population, but we haven't fully characterized, yes.
Okay. Maybe we can take
would like to continue?
Let's do that.
A question I have is also a question from Christine, and I have the same. You show us that, in fact, you have a different topology of tissue coming from the same biopsy. If you are performing different lymphoid, you have, in fact, different presentation of the tissue. So how do you take this into account? How many lymphoids did you need to have from the same patients? And to some extent, do you have sometimes a response to a drug in part of the biopsy, one lymphoid and not in the others? Is it something that is interesting, or it is an issue for you? How do you deal with that?
So for what we noticed that we need that we did different tests, at least 6 different lymphoid for condition. It's what we put to account for this heterogeneity. So when we chop the tissue, we randomize completely. So, and then we seed 6 random pieces. For the analysis that we have done in terms of response to therapies, we section multiple at different levels. So we go on multiple slides to make sure that it's not just one area that has a lot of response and the other one no. So we actually go through quite a lot in terms of slides that we analyze at different level of the tissue. And so the results that I show you is actually multiple points that we counted together as an average response that we had. So this is how we had so far addressed the question. If there is a response or not, we are not paying too much attention into that yet, unless we can really demonstrate that there is a different microenvironment or a different genetics. For the moment, we think that is the heterogeneity of the system, but it might be that is, it's interesting, but it's a bit, too early to speculate.
Okay.
I think we have another question also from Sylvain Lamure. Have you compared in your have you compared drug testing on dissociated cells in parallel to the lymphomoid to see whether the response was different, or what was the advantage to do it on lymphoid as compared to cells that you just dissociate and you put in culture?
I have to say I don't remember, so not regularly. So it's not something that we have regularly test. What we like it, I think there are different approach that can be used. So you can be a dissociation and try to reaggregate to form some, let's say, organoid models. What we like of this, it's try to maintain the physiological tissue architecture. Of course, might be that dissociating the tissue could get a similar response, but you might have lose who are really the cells that are interacting at that moment with the cancer cells. And of course, as we maintain the tissue, what we want to explore a little bit more, it's of course using spatial transcriptomics to actually see before and after therapy, how much does it change, creating a cohort that can give us a little bit more information. If we dissociate, we lose this possibility. So we haven't done a strict comparison to say this is better. I wouldn't say so, but there are some advantages on doing this.
And the dissociated cells might not be able to persist in culture for a long period of time, which is also a large limitation of that.
Yeah, we have a mix of normal and cancer, so there is a little bit of everything. So that is what we found interesting on that side.
Perhaps we need to have a more general question also, and if we have time, we'll come back to more specific question. But if we move to a general question, the two presentations were really amazing and really showing that all these models could be complementary. If we could have a kind of provocative question, what is for you, both of you right now, the place for PDX? Because you have these very nice in vitro models, we have these very nice mouse models with humanized targets and all these things. And then you have the PDX system that is somewhere in between. So could you, both of you, perhaps comment on that and give your feedback, your feeling about that?
I'll start. Yes, PDXs are very useful. First, it's patient material, so you can have all the heterogeneity of the patients. If you have a collection of lymphoma PDXs, for instance, myeloma PDXs, the problem of the PDXs is that we usually transplant them into immune deficient mice, right? So there's not immune cells there. But the second approach that we are developing, as many others, I think, is the humanization of the immune system. Using hematopoietic stem cells, you can have a humanization of immune cells, T cells, natural killer cells, even myeloid cells that can be there when the tumor is there, and this is allowing to test, for instance, CAR-Ts, T-cell engagers, any immunotherapy combination in human cells, tumor human cells, and immune human cells, which I think.
Should these cells come from the same patient as the tumor cells?
Ideally, yes, but technically it's very, very difficult, right? So what we usually do is to use at least two different donors, to doing in parallel 2 different experiments, sometimes 3, in order to be sure that we are the immune reaction and the immune therapy responses that we're getting there have sense.
Okay. Elisa, what is your feeling about that?
But I think the PDX models are useful to at least have some systemic response and fast response to therapies. I think we acknowledge the limitation, but for example, what we cannot do on having this patient material that we use, it's a long-term treatment and tumor relapse. So while PDX could still be useful in this sense, and I think if we combine with humanized one, of course, it's if you have a humanized system, then you have almost a perfect model. It just takes really long time, and that it's a little bit where we need to balance. But I think they are still quite useful models for drug development in particular. Yeah.
So maybe I have also a question regarding the mouse model. So what you have shown is, as an example, is a quite aggressive model with MYC in multiple myeloma, which the latency can be very short, or I suppose can be short. What we have as a problem in mouse modeling is in indolent lymphoma, where the development takes years, and also the latency is very long, and not all models are developing tumors. So have you succeed because in your approach to develop a more homogenized indolent model, or do you have some idea on that? Because this is, I think, something where this kind of drug treatment, where you don't know whether you have a tumor or not, is quite difficult. So we'd like to have your insight on that.
Yes, no, this is a very, very important question, because myeloma is a slow-growing disease, such as many indolent lymphomas. So in the mouse models, we're not interested in having these aggressive myeloma models, but also to have the slow models of myeloma. So indeed, we have a collection of not indolent, but myelomas that develop progressively, the precursor stages, and can get myelomas in 11, 12 months of age, which is a long time, but you can see this progression very nicely from month to month, for instance. And we've been able to treat myelomas, not only, for instance, at the myeloma stage, as we do in our patients. So before we have treated what we think is similar to the smoldering myeloma space in these slow-growing myelomas. Technically, they are very complicated to work with because it takes a long time, a very long experiment, very expensive. But I agree with you that the real myeloma or indolent lymphomas is the same scenario, are the best models, not those with MYC, very aggressive, but those without MYC, without these oncogenes there. Yes.
And sometimes they are difficult to follow also over time, because we don't have blood spread and etc. So yeah, but it's good if it's working.
Yeah, fantastic. So we have perhaps time for a few two last questions from the audience. The first one from Jean-Philippe Merliot, perhaps for Elisa. In your lymphomoid, do you have to face EBV positive bystander cell expansion, or I should say perhaps more generally normal B cell expansion that could overcome the development of tumor B cells? In particular, probably the question that is behind that is in the context of, again, indolent lymphoma or slowly proliferating lymphoma, how it works in your model.
I think the model is a relatively short time, so we don't see a lot of B cell expansion because we push the system to 4 weeks, but I think it's really the limit. The best condition are between 1 and 2 weeks, where you have really good maintain the environment. And so in this case, we don't see a lot of normal B cell expansion. So I think in this case, we kind of maintain a similar proportion between the normal and malignant cells. But because it's a short time, so it's not organoid really on.
Okay. And another question for you regarding your very interesting optogenetic model, again from Frédéric Moursin. How long can you keep the explants in culture?
So we push for 4 or 5 weeks now. But that's
That is quite good.
it's a good, and I think it's nice. We can see a little bit the progression over time. So it's really it's, of course, the mouse model has been done, so it's nothing new there, but it's really about the initiation. We see how these cells from the beginning to the end, maybe we can follow them. So
and also perhaps a way to homogenize, because in the mouse model, as Sandrine said, each mice is different, in fact. And so you don't know at one moment whether these mice, what is the stage of the disease, whether you have already a lymphoma or not. While here, starting from that, you could follow this perhaps more homogeneous, no?
Yeah, we started to do some single cell analysis over time and see a little bit how these cells change. So capturing really the beginning of the disease that otherwise it's difficult to capture either in the patient or in the mouse as well, because you don't know when actually the tumor starts.
And also what is good with this optogenic tools is that we are combining the mouse and
yes, amazing.
Yes. So I think it was a good
together, because it's actually they are good on engineering. It's much more easy to engineer mouse cells. So I have just one question for José that I found particularly interesting on this humanized protein that you put. But you create a full transgenic animals that express this extracellular part of the human protein to actually test different therapies, or you have maybe a shortcut strategy?
We have cell lines that have been mutant cell lines, humanized with all these proteins that I mentioned, which are very rapid to work with. But yes, indeed, we generated transgenic mice with extracellular humanized BCMA, for instance, that then are crossed to the myeloma models. And after a long period, we get this. Yes.
They're quite powerful, especially to test the response to specific therapies.
It's a long time. I think your experiments are much more rapid and very informative as well.
I have to say that in Switzerland, we have a strong regulation, so they push us also to find alternatives.
Yes.
So,
okay, so I think if there is no more question and we are a bit running over time, so I think we will have to close the session. So I would like first to thank José and Elisa for their time. They're showing new data that are very interesting, and I think I hope that the public enjoy also the two presentations. We would like to thank also the participants for their question and through this RedTalk. And so I think there will be a next session of the RedTalk on the 21st of May. So if you will receive some announcements, if you want to participate, I don't have the topic, unfortunately, but and so it's time to close. Thank you, Karin, and I think thank all for joining us today and for your active participation, and hope to see you the next RedTalk. Thank you.
thank you so much. Bye-bye.